Recall. Okay, so now we're going back to the eye. So last time was the quiz, which you did a great job on. And so now we're going back and we're gonna go literally as if we were following a photon of light. We went into the brain and then we go all the way back, and we're going to start from basically inner and then go straight back towards the photoreceptors. So okay. Here we are, and we're going to focus on amacrine cells. Okay, so let's see if I can. Okay, so again, that's the whole point of this, is so you can read that. So that's the most important thing of all these lectures, that you can read that. And again, I just show you, this is what, our OCT looks like, and don't be nervous, and always just realize that this is still complicated. Even if we go through all the lectures and you don't know what a layer is or whatever, just remember you're better than you were before. So we just keep getting better. So let's talk about amacrine cells, which are kind of like enigmatic, right? Because we just kind of reference them, but we don't really talk about them. But I actually think they are they're like the sort of metropolitan, worldly, sort of sophisticated cell of the retina, because there's so many of them and they're so different. There's over 40 types found in all living beings. They are interneurons, so there's still no difference. They're not like your Mueller cell or your RPE. Those are the two non-neurons. They're still helper neurons. But they also have different characteristics. Like some can be bistratified or monostratified, which makes sense because where they play in the inner plexiform layer, and the inner plexiform layer has strata, right? It's the one layer we keep talking about that have sublaminas, and they have, different strata. But the amacrine cells have different ways of designating themselves, right? Meaning the way we designate them is different. So first of all, there's one way of designating them, which is not really the way we use it, but it's good because it kind of jives with what we've talked about before. It's through their fields. And when we say their fields, all I'm talking about is what their field is for their dendritic tree that goes in and connects to other neurons. So how big is that? So this is a narrow one because you can see it when you draw it out as a narrow field, almost like a midget, but it's not called a midget. But it's like, that's the type of thing when we refer to fields, we're talking about the midget versus parasol kind of distinctions, where the actual dendrites have different kind of flaying out kind of characteristics. So this one's narrow, then you have a small, you have a medium, and you have a wide. Okay, so just know that amacrine cells are heterogeneous, and they kind of have these narrow and then wide fields, and can be really, really wide. 500 microns is massive, right? I mean, 500 microns is basically half a millimeter. I mean, that's pretty huge for a cell, right? So these are the types of things that you should keep in mind. But and then let me show you an example. Here's some monkey retina amacrine cells, and you can see, right, here's one that's really small, and then here's some in a cat, right? These are really wide, the large fields, because they spread out really far. So this is pretty easy to understand, right? And this corresponds to kind of this one, if you look at it from top down on the right, and then you can see from the kind of cross section here on the left, and you can see them. That's the amacrine cells. But mainly, I like to look at them from the class, like when we classify them, there's the size designation, but then there's this like class of amacrine cells kind of writ large that lets you understand functionality. One of the reasons why I keep stressing what is a midget cell and what is a parasol cell, and what is a midget bipolar cell and what's a diffuse bipolar cell, it's because it also implies functionality, right? Because if you're a midget cell, you've only got one connection, and that means something, right? You can start to think, okay, well, maybe it's probably one cone. That's what it's going to be. So the same idea here, if you classify them based on what's called their inhibitory neurotransmitter, that kind of gives you a sense of what type of amacrine cell they are. So there are two main types of neurotransmitter that is secreted by amacrine cells. The two main types are GABA, which you've heard of, and glycine. I mean, you know that these are neurotransmitters in the brain, but just know that, the only neurotransmitter you and I have talked about is what thus far? What cells? Glutamate, right? So that was the photoreceptor that secretes it when it is dark, right? So that's the only one, and that made it quite simple. We weren't going to talk about like different types of neurotransmitters until now, okay? So now you realize there's actually other neurotransmitters in the retina. Like if I were to section the retina and just like, spin it down and then separate out all these chemicals, you would see that some other neurotransmitters are being secreted, one of which is GABA and one of which is glycine. These are inhibitory neurotransmitters, and then there are 15% of all amacrine cells that have neither. They're not they're not secreting those two. But that's how we classify them. So remember this other thing, they're just inhibitory. So in other words, this is how we classify them as in terms of their inhibitory neurotransmitter. So you can secrete other things, but you have an inhibitory neurotransmitter, and in particular, if you're GABA or your glycine, that's how you would classify them, or neither of those two. But it's based on your inhibitory neurotransmitter. So as an example, we have an amacrine cell here, and then it's for inhibition, meaning of the downstream neuron it's connected to, it secretes GABA. Well, then it's a GABAergic amacrine cell, because that's what it secretes as an inhibitory neurotransmitter. But it can also happen to secrete something for excitation with another one of its arms, so to speak, its dendrites. And let's say that was acetylcholine, just throwing that out there. That is what it's secreting to excite. Well, it doesn't change. It's still a GABAergic amacrine cell. That's still what it is, despite the fact that it's secreting a whole nother neurotransmitter for excitation. That doesn't matter. Its classification stays because of its inhibitory neurotransmitter. Another one it could secrete is dopamine. Let's say instead of acetylcholine, it was dopamine. That doesn't change it to a dopaminergic amacrine cell, because that is not its inhibitory neurotransmitter. It's GABA. But then if I say, oh, I'm going to switch that one to glycine, well, now all of a sudden it's a glycinergic amacrine cell. Does that make sense?
Yeah.
Okay, so it's classified on the basis of its inhibitory neurotransmitter, but it can still secrete excitatory neurotransmitters as well. Okay, yeah. So there you go. Okay, so let's go in and just take 3. There are too many to count amacrine cells, and totally there will be, like, by the time I'm done with this lecture, there might be another one that they discovered in some species. So broadly speaking, we're going to talk about 3 just because it's good to just have something in your toolbox to say, okay, I know what an amacrine cell is. Here are the 3, okay? Here's one, and I like this one because it's actually pretty interesting. This is a GABA secreting amacrine cell. If that's the case, I just told you this, it's a GABA secreting amacrine cell. What is its inhibitory neurotransmitter?
GABA.
GABA, easy. It's not a trick question, right? It's this like idea, who is buried in Grant's tomb? It's Grant, okay? Like, it's the same idea here, right? GABA secreting amacrine cell. GABA is its inhibitory neurotransmitter, but we call this one the starburst, okay? It's a special type of amacrine cell. There are 2 types of starburst. We're just going to go into it so you get a sense of what it is. There's an A and a B, and now in your head, you should be thinking, well, I know there's an A and a B nomenclature that we've gone through, but, sublamina A, sublamina B. It's also off and on. And you're like, wait a minute, you can have an off on starburst? Okay, cool. Well, there is an off on designation you know of very well that exists in the IPL. So then let's draw it. This is a kind of one of these a classic amacrine cell, but this is actually how it looks. It has a bunch of little processes. And then where is it? This is how it looks in reality. And that's how it looks. I mean, these look literally like starbursts, right? I mean, they're beautiful. They're almost like in the sea, almost like a sea creature, right? So you look at this and you say, okay, well, let's see where they are. Here's your IPL. And then my INL is down below because I'm a clinician. I'm not a PhD person drawing this. So thus we have it oriented the correct way in our view. And so your IPL is there. There's the ganglion cell layer, and you have A and B because that's how we name things. And the distal one is on and the proximal one is off. This is review. And basically this is where the amacrine cells sit, like that. Okay, that's how they sit. And this one actually sits in S4. Do you remember how even the IPL, it's not just A and B, it also gets even more divided, like S1, 2, 3, 4, 5. Do you remember that?
Yeah.
Yeah, so this one sits in S4. That's where its like processes are. And this one sits in S2, interestingly enough. And who do they connect to? They connect to this guy. Well, what is that guy? Who is that?
It's a coniocellular.
And what type of cell?
Ganglion cell.
Ganglion cell, right? So now you are like I don't have to say anything, right, Jeanette? Like, I mean, I could look, you see this and you go, huh, you can already tell me what is the coniocellular pathway? What does it deal with?
Blue light.
Blue light as well as what?
Proprioception.
And proprioception, right? So this particular one, and we call this one A and B, which is great, okay? It's for proprioception integration. That's what these amacrine cells help with. But that makes sense, right? This would be the ganglion cell that it should connect to. You'd be confused if it was connected to like a parasol ganglion cell, right? You'd say like, okay, maybe that's what it has to do. I don't know, but it would be a little bit more consistent if it connected to the coniocellular pathway cell, which is the bistratified ganglion cell. So in this case, you have proprioception and integration. Now, Here's the interesting part about the amacrine, this amacrine cell, which is again is a GABA secreting amacrine cell. This is called a starburst because it looks like a starburst when you put it on a plate and you stain for it. Here's what it actually looks like. It doesn't actually look like this. Here's where this guy's nucleus is, and we call him displaced. And here's where this guy's nucleus is, and we don't call him displaced. Now, why is this interesting? Because this guy makes sense. This is his nucleus. He's in the internuclear layer, and I told you. The internuclear layer houses everyone, right? I mean, like we know that. That's what I always told you. Everyone, amacrine cells, mostly all the amacrine cells are in the internuclear layer, except this guy. Where is he? His nucleus is in the ganglion cell layer. He like slipped into the penthouse. So it's a interesting, and that's why they call him displaced. Like they needed to come up with a way to be like, wait, there's an amacrine cell nucleus here. We're going to call him displaced, even though he exists. And his nucleus is in the ganglion cell layer, which we always believe is only for ganglion cells. Sorry, trivia question would be, does the ganglion cell layer only have the ganglion cells? Now you know the answer that you were saying was correct, but incomplete. It also houses the displaced starburst B nucleus, okay? And of an amacrine cell. So it's actually ganglion cell 99.99%, 0.001%, amacrine cell, specifically this one. Isn't that interesting? I don't know. I find that interesting that it's called the ganglion cell layer, but now you know that, well, there's a little bit more to the story here. Now, this is actually, it's a interesting kind of cell. This is what it looks like. If you look in the human retina, S2, which is the one that's the lower kind of A version, the starburst A, that one is just called a starburst cell because his nucleus is in the internuclear layer. But B is the displaced starburst cell because its nucleus is displaced into the ganglion cell. That's why we actually call it that, because it's kind of like it shouldn't do that, but it has to because it kind of sits like this. So that's why we explain that. So I think that's pretty cool. It's neat. What is its purpose? Directional motion. Again, proprioception and integration. You don't have to know the specifics here, but just realize it will secrete, when it's activating something, it'll secrete acetylcholine. It deals with kind of different motion. And then when you have like centripetal motion, it's the inhibition of GABA. When it's centrifugal motion and it's going away, it is activation with acetylcholine. So it has these two arms. All you have to know is these starburst cells tell us that and relay that information to a ganglion cell, who then tells your brain something is coming at me or something is going away from me, which is very important if you're being chased by a lion, correct?
Correct.
Right. And so what I was trying to get at is that's that integration. That's that proprioception. You're starting to see kind of, not everything is just a flash of light, and not everything is just basic kind of, like a small photon or whatever. Sometimes it's like, I need to understand how things are moving. Well, we're doing some of that processing in the retina. It's not always just in your head. It's in the retina, and these amacrine cells are helping with that. In particular, the starburst one helps with this kind of centripetal motion. Like you can imagine, if you're a rabbit, you probably have some pretty good starburst cells, right? Because you need to run away and you need to know if it's coming at you or going, right?
Yeah.
And so there you go. So this other kind of weird kind of thing is acetylcholine's in the retina. It's being secreted in the retina. Well, starburst amacrine cells are the main source of that, probably because we deal with motion all the time when things are coming and going from us. Understanding how that's going, is it going away from me, towards me? That piece of information is probably ridiculously relevant on a constant basis. So it's constantly secreting acetylcholine sort of as that's happening. And so thus, if you were to biopsy the retina, you would get acetylcholine, like a lot of it, and you would know the starburst amacrine cell is the one that's secreting it. Would it be the photoreceptor? Yes or no?
No.
No, of course not, right? Because the photoreceptor doesn't secrete acetylcholine, right? And so there you go. So now you know that, right? So it's, again, glutamate is still being secreted by the photoreceptor. We're just talking about an amacrine cell now, and we know this one's GABA secreting, so it's inhibitory neurotransmitter. By definition, is going to be GABA. We know it secretes it. But it also secretes this excitatory one called acetylcholine, which is at like a muscle junction and stuff like that, right? Like that's what that's for, but it's also found in the retina. And when they first probably discovered this, they said to themselves, like, how is this possible? There's no neuromuscular junction. It's all neuronal tissue. Oh, it's coming from these guys. They're using acetylcholine in a different way. Does this make sense?
Yeah.
Cool. This is a nice movie of, I believe, a turtle, if I'm correct. a turtle's starburst, and this is its IPL and only standing for starburst. So look at how perfectly aligned. This is its IPL on cross section. If we do this, look, it spins. You see the lattice, right?
Yeah.
And then you spin here and you see it's like that. Do you see that? I mean, we'll do it again. I think that's unbelievable. And this is just the starburst cell. We're not talking about all the other stuff that goes involved in the IPL, but notice how perfectly aligned they are, right? Because they need that kind of alignment probably to understand centripetal motion coming and going. You can't really make that huge of a mistake there. Otherwise, whoever made that mistake probably got eaten or something. You know what I'm trying to say? Like, you know, it didn't like avoid the obstacle that was coming at it. They didn't evolve. And the ones that have really good GABA secreting starburst cells did a really good job. Okay. So that's that. Here are the types. I'm just showing you a ton of types. Ren. There are a ton of types here. So, so we have starburst A and starburst B. Notice something. Who is this drawn by? This is drawn probably by someone who is a PhD. Why do I know that, right? Because internuclear layer is up, right? It should be down, right? Internuclear layer should be here and GCL should be here. What's reversed, right? Well, guess what? Look at all the nuclei, internuclear layer, all in the internuclear layer. And then boom, here's your starburst B that's in the ganglion cell layer. That's why it's displaced. It's just kind of cool. Now you know if you ever see one of these, yeah, there's an amacrine cell that has its nucleus somewhere outside of it because it kind of needs to be localized directly in this horizontal space in that sublamina B. Does this make sense?
Yes.
Cool. So, okay, so that's one, okay? Starburst. Now let's go to glycine secreting amacrine cell. Now you already know this one. You just probably don't know you know it. This is A, and that doesn't say O, okay? That says A2, okay? You remember maybe A2, amacrine, I guess 2. I'm not sure if it was the second amacrine cell discovered, but A2, okay? We remember this one. It has a narrow field. This is what this guy looks like. Now, he looks like a squid, not like a massive octopus or something, right? He does look like an octopus, I guess, but it's like really small, right? Narrow field, right? It's not like your starburst, right? Which is really big. And so you say to yourself, okay, it's a narrow field, but we know it secretes glycine as an inhibitory neurotransmitter. We know this to be true, right? So you say to yourself, okay, well, let's figure out where it is. Well, you remember this whole schema, right? Do you remember this whole thing where light hits a rod, the rod transmits to the bipolar rod bipolar on cell, which is actually a diffuse on bipolar cell, connects to a bunch of rods. So then it gets that information, but these guys are not connected to a ganglion cell. What do? They relay their information to a nearby cone who is like sleeping. And when he's sleeping, his Z's are glutamate, right? Glutamate's being secreted. And what does that do? That makes the bipolar off cell active and the bipolar on cell inactive. But what does this do? It transmits the information to this red guy who is the amacrine cell. And then what does he do? He says, okay, I'm going to inhibit this one and I'm going to send the message through here. So he's hijacking this on bipolar cell using the cones on bipolar cell. Make sense?
Yes.
Okay, so we call that person A2, and this is what he looks like actually. I'm just going to draw him out. This is what he looks like. Here's your rod. Here's your rod bipolar cell. He has a connection to this person. He has a connection to this on, and then he has a connection to this off. He's connected to all of them. And then you have this line, and look, there's the IPL, and now we're kind of in the right spot. The IPL, here's the on bipolar cell, here's the off bipolar cell. They're terminating in the correct regions. Here's the on, right in the on layer. And then this guy kind of, his internuclear layer, that's where his nucleus is, because that's the only one that's special is the displaced starburst B. Everyone else does what we said. Everyone else lives in the internuclear layer. So this guy lives in the internuclear layer, but he's connected to all of these people. And then you have this guy A2, right? So that's how this works. A2 is key. Remember what I told you. If you knock him out, you cannot see at night, right? Does that make sense?
Yes.
Okay. So if you knock him out, you cannot see at night. And that's an important thing to know, okay? Because like, it's not just the rod bipolar cell, but it's also A2 that's involved, who is a glycine secreting amacrine cell. So one of the things I'd like to say is, notice his inhibitory like synapse is this guy, right? He turns this person, he blocks this person so that it's not secreting. Well, he's probably shooting glycine here. You get what I'm saying? Right.
Yeah.
And that kind of gets everything connected in your head, right? Like that's what he's inhibiting. But notice he has to excite this one. Now, I don't actually know what the excitatory neurotransmitter is. I just will be honest with you. I haven't looked that up, but we know that he can do both because these amacrine cells can do both. And that's the wonderful part about them. They can kind of help and do these relay messages with both. But we do know for a fact, glycine is being secreted to inhibit. And why do we know that? Because he's classified as a glycinergic or glycine secreting amacrine cell. Cool. So provides the scotopic response. Makes sense, right? Because it's the rod bipolar cell. Inhibits the cone off with glycine. It's also known as A2 or AII. And let's go find him. There he is. And it makes sense how they drew him. Here's his nucleus. And then he has some stuff in the off layer and then some stuff in the on layer. It makes sense that they draw him bistratified. Does that make sense?
Yeah.
Right? Because he's connecting with an on and an off. He's kind of like a bistratified amacrine cell, if you will. Make sense?
Mm-hmm.
Right? Cool. And then the final one, and I'm going back to GABA secreting, but you have a GABA secreting amacrine cell. And I'm only bringing this one up because I think it's cool. It's A18, which must mean it was the 18th amacrine. So, but why? Is because it also secretes dopamine. And that to me is just totally weird. I would never have expected the retina to have acetylcholine and dopamine in it, but it does. And it does because of these guys. This one, you already know this because I told you for free. It's a GABA secreting or a GABAergic amacrine cell. You already know. I don't have to tell you anything else. GABAergic amacrine cell means that's its inhibitory neurotransmitter, period. You already know that. And so we know this to be true just from the name, but then it's a dopaminergic A18 cell. So then that was specific. You say, well, that must be its excitatory neurotransmitter. Okay, there you go. And they're so wide, they ring around these A2 cells, these other, the ones you just saw for the scotopic response. So here is a picture, and you have this A2 cell that's inside, it's a narrow field, and this guy is like ringing around him somehow, like, you know, it's like doing something to him, okay? We know kind of what happens, but it responds to thyroid hormone. Now, let me ask you this, Jeanette, I'm like throwing this stuff, would you have ever thought thyroid hormone gets into the retina and does something? No, the answer is no. Isn't that crazy? And I'm just trying to emphasize this, that these amacrine cells are like, again, the metropolitan sort of worldly, they know all the things, they know different neurotransmitters, they respond to things, they try new things, they're wearing weird clothes. Like you don't necessarily understand what's happening, but this is like why this organ is so interesting, because here you have a dopaminergic amacrine cell. So we're going to kind of do it ourselves. Like, I'm just introducing this to you, but like you can parse this. You say, okay, thyroid hormone, it deals with circadian rhythm. And this is what they look like. That's A18 way up here, okay? It deals with circadian rhythm. So if it deals with circadian rhythm, doesn't it seem like it would have something in common with our other things that deal with circadian rhythm? And what is that? IPRGCs, remember those guys?
Yeah.
Those deal with circadian rhythm. They deal with, like your pupillary light response. They deal with things that are the suprachiasmatic nucleus. They deal with things that are not like that are autonomous sort of, right? And so you say, okay, well, thyroid hormone kind of fits maybe, right? It's like a basal secretion and it's secreting dopamine. It's kind of interesting, but in your head, you should be like, if they deal with circadian rhythm and it's this metabolism, basal metabolism kind of situation, responding to a hormone like an endocrine, like organ almost, like, which is just like, blows my mind that it even exists, but it has something to do with IPRGC. So then I just went, and I just checked online because it makes sense. If you said circadian rhythm, is there some connection between the two? And I read this: parallel inhibition of dopamine amacrine cells and intrinsically photosensitive retinal ganglion cells in a non-image forming visual circuit of a mouse retina. You can actually parse the entire thing, right? Parallel inhibition, okay, inhibition or excitation, maybe I don't know what that means. I mean, I know what it means, but I don't know how it relates, but of dopamine amacrine cells and intrinsically photosensitive retinal ganglion cells, that makes sense, in a non-image forming visual circuit, obviously, right? Because you already know that they're not part of the three. Makes sense?
Yes.
So when I do these lectures, I think one of the wonderful things is to say, look, the whole point is to get this OCT to, like, I can read the OCT, okay? But then when you go through PubMed or you're reading something and you see something like this, you're going to say to yourself, I think I can parse this, right?
Yeah. Yeah.
And I think, and like, for instance, this makes complete sense. Maybe you're like, I need to know what parallel inhibition is, but I don't necessarily, I'm not confused about these things. So if they had said the opposite, let's say they said, you know, this and this in an image forming visual circuit, you say something's weird here.
Yeah.
Right? Because you'd say that doesn't make sense. Maybe they've discovered something that is truly magical, that like there's another way to form images. And we ought to learn that because, you know, we lose people for RP, but maybe that's a mistake too. And I can actually adjudicate that upon reading the title. And I think that's cool. So I'm just going to go. Oh yeah, here's Amacrine cell functions in general. You know, I try to do like a summary slide here because, like, you know, Amacrine cells just have a generic kind of thing. There's so many different types, but they create these functional subunits within receptive fields of many ganglion cells. So basically they can take in a lot of information from different ganglion cells and creates these like unification situations. And why is it important? Because some of that processing that we expect to happen in the brain happens before that in the retina. And I think that's one of the things that we should just remember. They contribute to vertical communication within the retinal layers. They're a helper neuron, right? They're helping kind of connections between, there's other, you know, bipolar cells. You have that example, right? With the bipolar cell of the A2, right? A glycine secreting amacrine cell called A2 that is what? That is taking something from a bipolar cell and helping along with integration way up to the ganglion cell, right? It's helping with that. And so that's a very classic one. Then they have these extensive dendritic trees. They do some inhibitory surrounds, you know, because you don't have to know inhibitory surrounds, but you know that inhibition is an important thing with amacrine cells. Why? Because they're literally classified based on their inhibitory neurotransmitter. So obviously that's important, right? And so that's important to note that they can do this. And why do I bring this up? Because it's like the horizontal cell, which I keep trying to fuse in your mind. These two guys do this. They do this kind of inhibition of lateral like cells around them. They're the helpers, right? They're always the helpers. And then this idea, I mean, like, I think this is novel to you today because, or maybe it wasn't. I don't want to say that you didn't read this, but I mean, most people, like when I read it, it was novel, right? It carries out paracrine functions, meaning somewhere nearby, it's secreting a neurotransmitter that's going to affect some cell nearby, like release of dopamine and acetylcholine. And then you know that without our amacrine cells, we have no scotopic vision. We don't have it. It doesn't exist. Okay. So I'm like, this is a relatively short lecture, but I want you to go through some of the amacrine cell functions and let's see, you know, if we can connect them. It's okay if you get them wrong because I just introduced them, but we'll go through, okay? So scotopic vision, if I say scotopic vision, which of the three amacrine cells was that one? Do you remember?
Is it the A2 one?
A2, you got it. It looks like it's not. It's A Roman numeral one, you know, I, but A2. Which one deals with those bistratified directional ganglion cells where it does centripetal and centrifugal motion? Do you remember that one?
It's the starburst one.
The starburst one, exactly. And that's the one that's GABAergic. A2 is glycinergic. And which one is glycinergic? I just told you.
The A2.
A2, that's the only one that I told you that was glycinergic. Importantly, I mention it because that's A2, right? That's one of the most important ones. If you're going to remember one amacrine cell, remember that one because it's actually participatory in the visual pathway. How about GABAergic, which I gave you two? Can you name one?
The starburst. Okay.
And then who's the other one?
The A18.
A18, dopaminergic A18, but the dopaminergic is talking about its excitation or its excitatory function, neurotransmitter, not its inhibitory, but exactly. What cell does canonically or classically does lateral inhibition of ganglion cells? What cell would that be? We're talking about it up here. I'll give you a hint. It's right here.
In right general, here. Amacrine cells.
Amacrine cells. That's all I'm trying to. Generally, amacrine cells do this. Okay, that's what you want to know, right? What would it be for bipolar cells, or what would it be for photoreceptors? It would be, or actually, it'd be, I should say, photoreceptors. It would be what? It would be horizontal cells, right? We know that very clearly because we've done truth tables talking about that, right? So this is amacrine cells, kind of sort of doing the same thing with ganglion cells, but we don't actually have, you know, a easy truth. I can't give you like a circuitry kind of thing because it's so much information is kind of going on, as you can tell. Which one of these deals with circadian rhythm?
The A18 one.
A18 one, right? Because it's dopaminergic, wraps around the A2, really wide fields, response to thyroid hormone. I mean, again, I just think it's just ludicrous. I mean, I think it's amazing, but I just think it's amazing that that exists, right? Who secretes acetylcholine and is responsible for the acetylcholine in the retina?
The starburst.
The starburst, exactly. Now, again, why do I bring up acetylcholine, right? Because we are not in a neuromuscular junction, right? And that's where acetylcholine typically is secreted. But now you know the answer. Someone says, well, therefore, acetylcholine can't be secreted in the retina. You're like, wrong. An amacrine cell does it because it uses acetylcholine in a different manner. In this case, centripetal or centrifugal motion. The displaced starburst, which one is it, A or B? Do you remember?
It was.
Do you remember? So remember where they are. If you're displaced, it means you're in the ganglion cell layer. That's why it's called displaced, because its nucleus is in the ganglion cell layer, and it's either A or B. And A is where? A is closer to the inner nuclear layer. It's B, exactly. So it's B, sublamina B. And actually its processes are in S4, but its actual, nucleus happens to be in the ganglion cell layer. Which one of these cells is dopaminergic of the three?
The A18 one.
A18 one, exactly. And what that dopamine does, we're not entirely sure, but it does secrete it, and it does respond to thyroid hormone. Which of the ones that I just described to you had a narrow field?
A2.
A2, exactly, right? Because the other one, starburst, was really wide, and it makes sense. You got to be really wide probably to see centripetal and centrifugal motion, but then also A18, for whatever reason, needs to touch a ton of A2 cells really wide. So the A2 cells are really narrow. They focus on that connection between that one or that diffuse rod bipolar cell and then the nearby cone on bipolar cell and nearby cone off bipolar cell. Which ones are wide then? What's the inverse of that? Which two?
The starburst.
Starburst and
the A18.
And A18, exactly. That's it. Okay. So that was amacrine cells. I will just tell you that the amacrine cells is like the zone in which if I were a betting man, I would say somewhere along the line, someone will talk about the amacrine cell. There'll be a new one, or someone will be able to manipulate one where it really changes like how we see. I mean, it really, maybe there's a regenerative function to ganglion cells, but I think that's where some of the space will start to alter, because there's so many different things you can do. Like you can see, it's almost like maybe I'll look in the amacrine cells to figure out whether there's a special like tool that I could use, right? Anyway, so that's amacrine cells. And next time we're going to do horizontal cells. Cool.
Okay. Wait, awesome. I have a question.
Please.
I got kind of confused when you were showing the connection between the A2 and like how it interacts with the photoreceptor.
No, so A2 doesn't interact with the photoreceptor.
You were talking about the horizontal cell.
Then. I was talking about the horizontal cell then, yes.
Okay.
Yeah, so like, let's just do it real quick, right? So here's your
that one slide, yeah.
Yeah, so where this is not it. So who is this? This nucleus I'm drawing to let you know that it's in the inner nuclear layer. Okay, right. That's where its nucleus is. Its processes are touching what? The bipolar cells.
Okay, yeah, those are okay.
Makes sense? Remember, this is a cone and then the bipolar cell on top of it. This is a rod, and that makes sense. But good question, because this same kind of drawing could be drawn if you flipped it around, if you literally just spun its things around and touched like a bunch of photoreceptors, then I renamed this horizontal cell. That's what it would be.
Okay.
Makes sense?
The bipolars.
What's that?
It's touching the bipolars.
Then. Right now it's touching this one is touching the bipolars, exactly.
Okay.
Cool. Because like if you were to spin this around and drop it down here, the layer would be the outer plexiform layer, right? And then these guys would be touching photoreceptors. So the problem is here, I'm not drawing in the outer plexiform layer. So it almost looks like this guy's in the outer plexiform layer, but he's not, right? It would be like smaller, right? Does that make sense?
Yes.
Cool. Yeah. So you got it. So there you go. And this guy's A2 again, he secretes glycine. Cool. Okay. So next time we're going to do, like I said, horizontal cells. Cool.
Yes.
Oh, let me just stop recording here.